I last week and I guess this week until Tuesday I'm in Boston again and then I'm heading home after.
We said when?
Right now until Tuesday.
Got it. In Boston, we're saying.
Yeah.
Cool. Any new full lengths that you've taken?
Yes, I took one yesterday and I put like all my information in the um Google sheet.
Full length two, okay.
I was happy that my chem physics score increased, but overall scores stayed the same again. Third time.
You said that your Chem Phys went up and the BB went up.
Biochem went down one from last time, I think.
Last time was underneath. Okay, got it. And your Psych Soc went up, you said?
Psych stayed the same as the last one that I took, I think. And then cars went down. So I just this morning I like did all the questions that I got wrong again for each section, but I haven't looked at anything past that yet.
Got
So
it. I see yeah, so I see on the free scored on the 18th, you got a 129 for BB. So or wait, yeah. Okay.
yeah, and this time was 120. I don't remember, 127, I think.
Yeah.
Or 128.
hmm Got it. Okay. So yeah, you did your blind review and your regular review. And do you have any questions from that stuff?
Well, I've only done the blind review so far, so I haven't had a chance to like go through the passages, but there's one I did notice that there was one chem physics passage where I got every question wrong. So maybe if we could go over that one. Um, Let me just pull it up. Okay. Um, this passage, I got all of them wrong.
Okay.
So I haven't looked at why yet. Like just looking at this problem, I think I know why, but yeah, I think
Okay. So I'm gonna have you read it out loud and tell me when you see those buzzwords.
okay. Three phosphoninositide dependent protein kinase 1, PDK1, phosphorylates specific serine or threonine residues in the conserved activation loops, T loops of other serine threonine kinases, resulting in the activation of those proteins.
Okay.
So I would probably highlight that it's a kinase and that it phosphorylates serine or threonine residues.
Tell me everything you know about kinases, because that's a very high yield topic.
Kinases add a phosphate group onto molecules and a phosphatase, I believe, removes the phosphate group.
What's what does pyruvate kinase do?
It adds a phosphate to pyruvate.
So if you go from oops, from pep to pyruvate, you use pyruvate kinase. And what's pep?
Um, it's oh,
Hmm.
um,
Does pyruvate have a phosphate?
no.
So
So it removes.
it can remove also. Yeah. So do you remember what enzyme class it falls under?
A transferase.
Transferase, right? Which means it's responsible for transferring phosphate groups. So
Mhm.
it doesn't mean that it always will add it to the substrate, right? It can, I mean, like if we had, glucose into glucose 6 phosphate, we're using hexokinase to take a phosphate from ATP and giving it to the glucose here,
Mhm.
ADP. So you could have that, but you could also have so PEP is phosphoenol pyruvate. So in this case, we're losing a pyruvate, right? So pyruvate is gonna sorry, the phosphate of PEP is gonna go to the ADP to produce ATP.
Okay, so like the kinase portion of the enzyme is like a kinase because it's like transferring the phosphate onto the ADP.
Yep, so it can either transfer a phosphate from ATP to a substrate like it does here,
Mm-hmm.
or it could take a phosphate from a substrate and give it to ADP to make ATP, which is what happens here.
Okay.
Now, so it can cause phosphorylation of stuff and also dephosphorylation of stuff. Now, they must use so are you able to like draw like what a phosphate looks like a phosphate molecule?
Um, like with the three phosphates surrounding
Like the three what? Like let's say we have a phosphorus atom here. What?
Mhm. Um, like oxygen,
Okay.
like four, I think four oxygens, is it?
And how should I write the bonds?
Um, is it like is there resonance like between yeah, the double bond with the other oxygens?
So now instead of drawing the entire thing again, I'm just going to draw a P with a circle, P with a circle, and then like adenine.
Mm-hmm.
Would this molecule would be what?
ATP.
Good. Now kinases. I mean, they're kind of helping us out by telling us serine and threonine, but you should know this that kinases must use either S, T, or Y. What do these have in common?
They have a hydroxyl group.
All right. And what do I say what do I say that you should be thinking of when you think when you see a hydroxyl group?
Um, like the ability to hydrogen bond.
So, uh, so whenever you see an OH group, uh, here's an example. So, what am I drawing here?
Glucose.
Okay. And is this like what most of the glucose in our bodies looks like?
Um, uh, well, there's two options, either having the end oxygen like on the right or the left side.
Okay.
Is that what you're asking?
Um, so
Or like in like the cyclic structure.
so um so it just so happens that one, two, three, four, five, six. One, two, three, four, five, six. So for the longest time, people, scientists and stuff, like I'm talking about like probably like 100 years ago, maybe even more, they knew that when it came to hydrocarbons, that stability like decreased as you add carbons, and then when you get to six carbons, it suddenly gets super stable. And then when you go beyond six carbons, it gets, less and less stable.
Mm-hmm.
So people always wondered why. And do you know the guy Kekule?
Sounds familiar.
He had a dream of an Ouroboros, which is a snake chasing its own tail. And he came up with cyclohexane.
Mm-hmm.
And that's why a six carbon hydrocarbon is so stable.
Mm-hmm.
So this is that going to be a very, very stable molecule?
No.
Good. Um, it would be more stable if it was a ring, right?
Yeah.
Now, how can this turn into a ring?
The hydroxyl group of carbon five, is it one of carbon five, like would attach to the carbonyl or the oxygen of the or yeah, the carbonyl carbon.
Uh-huh. So, what is it acting as, the hydroxyl group here?
Um, is it a leaving group
Uh,
or
it doesn't seem to be leaving. Seems like it's making a bond, right?
yeah. So like an electron donor.
Um, Something else. It
Uh,
so it there's electron donating, which could be strictly donating electrons, and then there's like an atom coming in, which has electrons, but you're also having the atom come in. But this is a acting as a nucleophile, right? Now,
mhm,
I want you to remember this. So I'm going to write it like a red star here. But I want you to train your like brain to whenever you see a hydroxyl group, think nucleophile. Okay. And whenever you see because that's the most common nucleophile that you'll see on the exam. Um, and yes, a nucleophile will donate its lone pair because
mhm,
a Lewis base is a nucleophile, right?
yeah.
So now your most common electrophile will be anything with a carbonyl group. That's because a carbonyl, so like a carbonyl group has a partial negative here, a partial positive here,
Mm-hmm.
and I'll write like ROH, um, has like a free lone pair. And so, and we call it a nucleophile because nuclei are positively charged, right?
Mm-hmm.
So it likes things that are positively charged or partially positive. So this attacks that electrophile like that. Okay.
Got it.
Now,
Yeah.
yep. So now, by the way, so only 0.1% of the glucose in our bodies is in this straight chain form. Um, Mostly all of it is in the ring form.
Mm-hmm.
And when this happens, when this nucleophilic attack happens, it makes the two anomers, the beta anomer and the alpha.
Mm-hmm.
Um, and the beta one is like 64% of the glucose in our bodies, and like 36% is the alpha one. But okay, so now STY, they all have hydroxyl groups in common, which means that their hydroxyl groups can act as nucleophiles. And for the same reason that a carbonyl is electrophilic, look at this. This is a little similar, right? You have a instead of a carbon double bonded to an oxygen, you have a phosphorus double bonded to an oxygen, as well as the phosphorus being bound to three other oxygens, right?
Mm-hmm.
So this makes it quite or this makes it the phosphorus have a pretty strong partial positive here, right?
Mm-hmm.
So the OH group of like a STY can attack this phosphorus and let me use like green. So attacks the phosphorus and form and whenever a nucleophile is attacking, it forms a bond. So it pushes this bond up here, brings this back, and then breaks this off. So you get that entire phosphate detached from the ATP. And as we move outward from the adenine, we can number the phosphates. Like the closest is alpha, then further out is beta, and then this is gamma.
Mm-hmm.
So you should know that. So you should know this about kinases. You should know that it's a transferase, which means you can't like pigeonhole it as something that's just adding phosphates or removing phosphates. It can do either because it's a transferase. It requires STY, um, the STY amino acids because they have the hydroxyl nucleophile. And
Mm-hmm.
if they are taking a phosphate from ATP, it will involve the hydroxyl group of the ST or Y nucleophilically attacking the gamma phosphate and pulling that back or pulling that out and giving it to a substrate, or it can take it can nucleophilically attack um, the phosphate connected to a substrate and then give it to ADP to make ATP.
Okay.
So yeah, just remember like just yeah, whenever you see your OH group, you're going to think nucleophile and carbonyl groups. Phosphate is an example, could be an example, electrophile.
Okay.
All right. So, okay, continue.
These other kinases possess a conserved PDK1 docking site known as the hydrophobic motif, which, when phosphorylated, interacts with PDK1. So I would highlight the fact that the docking site is known as the hydrophobic motif, and it gets phosphorylated and like would interact with PDK1 only after it was phosphorylated.
I also want to ask you, like, what do they mean when they say conserved?
Um, I mean, conserved activation loops, it's not um I don't know, like it's still there, like it's kept.
So when they say conserved in this context, meaning it's so this specific PDK1 um phosphorylates specific serine and threonine residues in the conserved, like whatever, activation loop of other serine threonine kinases.
hmm
Conserved just means that it's throughout its evolution, it's conserved or kept like stays, in has a lot in common with these other types of kinases in the kinase family because they kind of evolved together.
Okay.
Mhm. So so that's conserved. So that's why they're saying that this PDK1 has these residues that are conserved in other kinases. So they, evolve together or have, this is a similarity.
So it's like a similarity to typical kinases, is what it's saying, or
Oh, just I guess the like particularly like I guess they're not telling us exactly, um yeah, it's going to have some like structural elements that are common to other kinases. So,
okay.
um yeah.
Okay. Um, Crystal structure studies have revealed that PDK1 binds PHM through a mainly hydrophobic HM binding pocket called PIF. PIF. Um,
So, so yeah, I was just thinking about the evolutionarily conserved thing. And so it's like the coronavirus would have like originated from like either like bat coronavirus, pangolin and stuff like that. I actually did like a bioinformatics like kind of project on this and compared like the genes and all that stuff. I found the biggest similarity between it and so the conserved is like so there's the coronavirus which is from like the bats and there was like SARS. I think that was from like I don't know. There's like the avian flu, which is from birds, like the swine flu, and stuff like that. So these viruses and they share a common evolutionary like heritage. So they're gonna have similar conserved residues. Yeah, conserved like yeah residues or sequences and stuff. So like so like COVID has parts of it that are conserved across like the bat version of it and all of that stuff. So,
okay.
Mhm. But yeah, so okay. They said these other kinases possess a conserved PDK1 docking site known as the hydrophobic motif, which when phosphorylated, and we call that PHM, interacts with PDK1. Um, What would phosphorylating something that's hydrophobic do to it?
It would give it a hydrophilic region because it would have a negative charge.
Very good. All right, cool. And yeah, continue.
Um, oh yeah, the whole sentence with PIF, and then PIF is situated
Oh,
adjacent.
sorry. I thought we were up to like the crystal part, or
Oh,
maybe
I read that one already.
okay. Let me see. Crystal structure studies have revealed that PDK1 binds PHM through a mainly hydrophobic HM pocket called PIF. Um, Okay. So I guess although, you could have a hydrophilic region.
Oh, I think you froze. Hold on.
Yeah, you froze for me too. Okay, you're back. Am I back?
Yeah.
hmm Oh yeah, yeah. So PDK1 binds PHM through mostly a hydrophobic binding pocket called PIF, okay?
hmm PIF is situated adjacent to a positively charged ATP binding pocket, but away from the catalytic site. Um, I would highlight that it's right next to a positively charged binding pocket, and it's not at the main like catalytic site. Um, so like I guess like an allosteric site.
Okay.
Organization of the catalytic site increases in PHM-bound PDK1, while no dramatic structural changes are induced upon T-loop binding.
Okay.
Yeah, I guess like I would say I would keep in mind that like organization increases like the PDK1, but
What do you think they mean by organization?
um like less flexible, like if it's more rigid, more organized.
I think they just mean so if they're saying like organization of the catalytic site increases PHM bound PDK1 while no dramatic structural changes are induced during T-loop binding, maybe they mean structural changes
Yeah.
with organization. Um, so okay, we have a bunch of kind of like things that are interacting with each other. I don't know in which direction they interact yet. So, I guess I'll just continue reading, or yeah, you can continue.
To better understand the mechanism of PDK1, researchers performed in vitro enzymatic and kinetic analysis of the enzyme. Um, a synthetic peptide with the amino acid sequence, and then they give you the sequence, was generated as a mimic of the T loop,
Okay, so they're doing this experiment. What's in vitro mean again?
like outside of the organism,
Yep. Like a test tube kind of thing. Okay. What's inside of the organism?
in vivo.
All right, now we're given a peptide sequence, and what I would do is I would find the charge of it. So, um, so yeah, what could the charge be here?
Well, that was actually the first question that I got wrong, but the only one that I messed up on was cysteine because originally, like, I knew that cysteine was uncharged, but then, like, I saw somewhere, like, giving the charge of the side chain. So then I like, gave it a charge when I was calculating the net charge, and that's why I got it wrong.
Uh-huh.
Um,
Let me see. So K is -1, T, F, C, G, P, E is, wait, no, K is +1. Um, then E is -1.
mhm.
Is that it?
Yeah.
So 0, right?
Yeah, so I just want to double check now that like cysteine does not have a charge,
Uh,
right?
correct. The ones that so like the way that I go over amino acids with students is like the three kind of degrees of specialness. So the first one is fully charged amino acids. So
Mm-hmm.
that includes the two acidic ones and the three basic ones.
Mm-hmm.
Acidic ones will be negative. Basic ones will be positive. Histidine's a little weird though, but those are the ones that'll be charged. Second degree of specialness is if the first degree is charge, second degree is partial charge. So that includes any polar molecule. But instead of like memorizing it as this is polar, this is nonpolar, you should have the ability to look at a molecule and determine if it's polar or nonpolar just from looking at it and looking for, there's a highly electronegative atom here, and that causes polarity and stuff. So that's going to include STY, okay? They all have the OHs. Um, Y is tyrosine, which has a phenyl group, which is hydrophobic, but it also has that hydroxyl group on the phenyl. So,
Mm.
and then you have your, so you have your SCY, and then you have the amide versions of the acidic one. So glutamic acid, turn that into an amide, you'll get glutamine, and then aspartic acid, you turn that into a amide, and you get asparagine. So STY, and then the amide versions of the acidic amino acids will give you the five partial charged, aka polar ones, and then the rest of the 10 are like non-polar. I mean, there's going to be certain exceptions and stuff sometimes, but yeah, just know that your your two acidic ones are going to be negatively charged. Your three basic ones typically will be positively charged except for histidine. That's the weird one.
Okay, so cysteine is never like its side chain is never charged.
Uh, at like physiological pH and stuff, yeah, it's not going to be, but yeah. Okay, cool. So yeah, so whenever we see an amino acid sequence, we can just kind of quickly like write out what the charge is on that because they're not going to really ask much else about that. And you could do it like when you have a question on it, but you could see right here that it's not really doesn't take too long to do it, right?
Mhm.
Okay. And yeah, okay, you can yeah, continue.
This synthetic T loop, ST loop, was incubated with 32 phosphate labeled ATP or phosphorus labeled ATP in the presence of PDK1 for different time periods at 37 degrees Celsius and pH 7.2, and the amount of radioactivity incorporated into ST loop was measured by detection of beta decay.
All right, what are some buzzwords here?
Um, well, I guess the phosphorus labeled ATP and the temperature and pH, and then the beta decay.
What can you tell me about beta decay?
Well, the beta like molecule that what is like leaving the molecule. So, it's like when you draw it out, it's like beta with 0 and then -1 at the bottom. So that would go like on the products side, I guess.
So a beta minus particle is has like what mass, what charge?
It has 0 mass and -1 charge.
Mhm. So it's like an electron, right?
Mm.
Um, what other decay products are there?
Alpha decay and gamma decay
Anything else?
and forget what it's called, but like the helium one.
Uh,
Or positive, or is that alpha decay?
so it's closer to alpha.
Okay.
So alpha, yeah, tell me about alpha.
Um, well, it has a 4 like mass and then a 2 charge. like helium. So
Is it
when
is it equivalent to helium?
no,
So how is it different?
um, I'm not sure. Is the charge different?
That could be it because is an alpha particle charged?
Yeah,
Is helium charged?
no.
So they have the same mass, and have the same like atomic number, but why is one charged and the other not charged?
Um, does does helium have like the protons to balance out the electrons, whereas the alpha particle doesn't?
Doesn't have the what?
Oh, protons?
So let's see, right? So what charge would an alpha particle have? Like positive or negative?
Negative?
Um, so
Positive?
so if like if an alpha particle has a mass of 4 and a, let's say, atomic number of 2, that means that there must be 2 protons and there must be 2 neutrons, right?
Okay.
Now, for helium, same thing here. However, for the alpha particle, we instead of just, well, we don't even really call it the atomic number, we just call it the charge. So the atomic number is the number of protons. It is equivalent to the number of protons. So both of these have the same number of protons, the same number of neutrons. However, the alpha particle is positively charged and the helium is neutral. So they must have a different number of what?
Electrons?
Yep. And how many electrons should helium have?
Two.
Okay. And if it loses two, it would have what charge?
It would have a positive charge.
Positive what?
A plus two charge.
Good. So yeah. So a lot of times when I ask students, like, okay, what's the alpha particle? They just say helium, but it should be the helium nucleus
Okay.
because a nucleus is always positively charged, right?
Mm-hmm.
Good. And so there's that, and then there's beta plus decay, which is a positron. So that's like um it's essentially like a positively charged electron. It's a mass of 0 and charge of +1.
Mm-hmm.
But okay. Beta decay, good. And all right, yep. Uh Continue.
This experiment was repeated in the presence of a synthetic peptide that mimics the HM domain, SHM, of serine, threonine kinases with the amino acid sequence. And then they give us the sequence.
All right, so we have another one of these. What's the charge of this guy?
0.
Yep, doesn't seem like any special amino acids in there. All right, cool. So now, all right, so they used, hold on, they used this synthetic peptide as a mimic of the T loop, and then they made this other synthetic peptide that mimics the HM domain of the serine and threonine kinases. Okay, and yeah, continue.
Phosphorylated SHM, SPHM was also used in place of SHM. Um, the effect of the addition of two potential inhibitor compounds, compound 1 and 2, to the PDK1 ST loop SPHM solution was also tested. And then from the measurement of initial reaction rates, the kinetic parameters Km and Vmax for each substrate were determined.
All right. So, yeah, so far, kind of hard to understand what they're kind of doing here. There's a lot of weird terms like T-loop and stuff like that, but whatever, we do the best that we can. So let's see, table one, kinetic parameters for PDK1 dependent phosphorylation of ST loop. So when you do this test, like I'm guessing you like skip the table and you go to the questions, then if you need to go back to the table, you go back.
Yeah, I looked at it. I usually look at it like, or I've been trying to look at it a bit more before going to the questions, um, and seeing like if there's any differences between the variables, like between the controls and then like compound one, for example, um, for KM at least.
Yeah. So I would say on the test, spend like 15 seconds to get the gist of what they're trying to show you in the table. And that, if it past the 15 second mark, just continue moving on with the passage. And if you need to go back, uh, they'll tell you. Uh,
Hmm.
and sometimes you don't even need to go back. So now when it comes to evaluating these, I always favor like a slow systematic approach. So that way just to minimize mistakes. So I would just kind of do like left to right. And of course, this is more easily done when you're doing your blind review. So okay, ST loop, KM, okay, KM, V max, um, control, some like 8 and 20, okay, SHM, which is the phosphorylated hydrophobic do motif. And in that, the KM, okay, the KM and Vmax stay the same. And for the SPHM, which was the phosphorylated SHM, what was the SHM again? Yeah, SHM.
The HM domain,
HM.
like right above. Yeah.
HM domain. Wait,
The like hydrophobic motif.
the hydrophobic motif, the S, is that the synthetic?
I think the S is the HM domain, and then the P is when it's phosphorylated.
I think, yeah, I guess the ST loop perhaps,
Oh, maybe, yeah.
or the
Synthetic,
synthetic peptide,
the synthetic HM domain versus the actual domain. Like, I think it's just coming from like this.
okay. So those results seems to have an same KM, but a larger Vmax. So the rate gets faster when you phosphorylate the SHM domain. So,
Hmm.
okay, so I think what they mean by so we know from here that the HM domain is HM, and we know that it can be related to make a PHM. But here, when they talk about the synthetic peptide, yeah, I think you're right on this one. The synthetic peptide that mimics the HM domain is SHM. So, and then that can also be phosphorylated. And the phosphorylated synthetic peptide mimicking, the HM domain seems to have a higher Vmax. And then compound 1 and 2, they told us were inhibitors, and we're likely going to be able to tell if it's like a competitive or not based on Km and Vmax. So let's see. Km goes up, Vmax is largely the same. So compound 1 could be what type of inhibitor?
Um, Km goes up. Competitive.
Uh-huh. Um, and what is Km?
The concentration at which
Concentration of?
the concentration of substrate at which the enzyme is at half of its maximum velocity.
Good, perfect. All right, cool. So yeah, compound 1 is, a competitive one. Compound 2, we have Km unchanged and Vmax decreased. So what can that be?
Um, that would be non-competitive.
Okay. Um, so yeah, okay, good. So, all right, let's see. Oh, you can hide these solutions. Um, okay. Oh yeah, oh, so this is what, yeah, okay. That's
Yeah, so this was just like the cysteine
yeah.
issue.
So you're saying that when that you thought cysteine would be what charge?
I thought it would be negatively charged from the side chain just because I recently was looking up the side chain PKAs and like it had a PKA so I thought it was charged.
Say what you said about the PKA again.
Wait, hold on, let me see. Um like think here. Yeah, like cysteine has like a side chain pKa, so I thought that meant it would be negatively charged um at physiological. But I mean it's also in the polar uncharged group, so I think that kind of messed me up.
Um,
Because when I was looking at this, all of the other side chains that had a third pKa, they were all the charged ones. Or I guess except tyrosine, so I don't know. Um, yeah, so I was just a little confused.
got it. So you're saying that okay, so um that side chain 8.35. So I guess if the pH is so okay, if we look at this, if our pH is less than this, it'll stay protonated, right?
Yeah. Okay, yeah.
And if it's, larger than that, um, it'll become uh deprotonated, right?
Mm-hmm.
Like I'm comparing like let's say I had something with a pH of like something, and I'm taking the entire cysteine molecule and I'm putting it in. So if it's a pH of like uh 7, then the environment is more acidic than the cysteine side chain, and therefore the cysteine side chain will get the environment that's more acidic will donate protons to it, so you'll get your CH, your SH, your thiol group. Um, but if you're putting it in an environment with a pH of like let's say 9, the environment is more basic, and the cysteine could then be taken can be deprotonated that way.
So in that case, it would have a negative charge when the hydrogen leaves.
Uh-huh.
Okay.
Yeah. So because pKa values you know, stand for like I guess the amount uh that is needed to either uh like let's say like deprotonate something or protonate something.
Hmm.
So so that's why uh yeah, so cysteine has this side chain of 8.32, but in physiological pH, that environment is going to be more acidic, and so cysteine is going to stay its side group at least. The thiol group is going to be protonated and therefore neutral.
Okay. Have you seen questions where like they ask you to find the net charge when it's not at physiological pH, or is it typically at physiological? Like, would there be a question where like it's at pH of 9 and there's cysteine in it and like they're trying to trick you because it's uncharged technically?
Uh-huh. So yeah, so I guess they could, I mean, I don't think they'll ask anything like that for like cysteine, but they can ask you to like um or at pHs that are other than physiological pH. Generally, no. Um, they could ask you about like finding the isoelectric point of an amino acid, and that means that, I mean, for non-acidic or basic amino acids, it's straightforward. It's just the average of the C terminus pKa and the N terminus pKa.
Mhm.
Uh, but if you have a side chain that's charged, meaning either acidic or basic, you're gonna do the average of the closer pKa's. So like for example, arginine, right? Side chain is has a high pKa here. And
Mhm.
what is it closer to? This or this? It's closer to this, right? So you take the average of this 9 and the whatever 12.5 to get like 21 or whatever, divided by 2 to get our I or isoelectric point of 10.76. Whereas for something like aspartic acid, where your side chain is small, you take the average of this and this to get your isoelectric point to be that.
Okay.
So yeah, so I don't think they'll ask you about like, okay, what cysteines charge, or this peptide at like pH of 8 or something like that, but they could ask you stuff about isoelectric points.
Okay. All right. So then in that case, we could skip that question.
Mhm.
Um, this one I got wrong because I wasn't really thinking through the question and assumed that they were talking about like the beta decay when in reality they were talking about which like which phosphorus is labeled specifically. Um, so that got better on the blind review, but
Yeah, yeah. So that's what we talked about with the kinases, right?
yeah,
How it takes the gamma phosphate, right?
yeah,
So, but yeah, like people would have chosen this because that's if you do the thing that I always say that people do, right?
yeah,
I always say that people, like like people always tell me like, okay, it's not going to be A because they never mentioned anything about alpha. Not going to be, they never mentioned anything about gamma or delta, but they mentioned beta and they, mix and match kind of thing.
yeah.
So those will always be your trap answers. So always be aware of that.
So do you think, like, strategy-wise, you should always assume a question is pseudo-discrete first before thinking about applying the passage?
Uh, well, you'll have pseudo discretes pretty much only in the chem phys section, right?
Okay. Well, like for this example, like it's a pseudo-discrete kind of like because you just have to know like which which phosphorus is the last one.
I see what you mean. So like, yeah, it's like I see what you're saying. So they're asking us about the like which phosphorus was labeled, and so we can use our knowledge of kinases, taking out the gamma phosphate. So I guess this could be considered that. Um, but to answer your other question, like the other sections are not going to have like too many pseudo discretes. Chem Phys is the only one that has some pseudo discrete stuff, but this is still something that we needed to like, read the passage for. Um, but yeah, I would just say to yeah, just start with or it's just a better mindset to have to think about connections to the passage.
Okay.
But you can have some pseudo discretes in Chem Phys for the most part only.
Okay. Um,
Okay, so it's like so yeah, so it's a good habit to do what I said to do when you see these amino acid sequences. Um, so SPHM they mentioned was
uh,
that thing, right?
yeah, like here. Yeah, I got this wrong because I was looking at the like sequence and threonine was like polar uncharged. So I was looking for a polar uncharged substitution when that's like not what the question was asking.
Okay, let's see. So, oh, you know what? This is a bit more here. What they're saying.
Yeah.
Like, what are they? Okay, so how can we?
Like they're asking um if you used SP or if you used a sequence instead of SPHM completely. And so you would need a sequence that already had a negative charge
All right, why?
because SPHM is the phosphorylated version of the SHM. So you would need a negatively charged amino acid if you're not using the phosphorylated sequence anymore.
Good.
So
This
I got that on the blind review, but I didn't get that on the original question.
Did you choose something that had a charge of 0?
Originally, I think
I don't even know if they have one that has
I
that.
think I chose B because
Uh-huh.
glutamine is uncharged, but it's polar.
Mhm.
And so I did that instead of like the threonine.
Got it. Got it. Yeah. So yeah, this is something called phosphomimetic, like glutamate, aspartic acid, right? These are having negative charges, so they can mimic the effect of like a phosphate.
Okay. Yeah, I think that was like a concept that I didn't like connect the dots to the first time around.
Yep.
And then this one.
Yeah, this is also like what I was telling you about how important it is to understand that hydroxyls, because they're asking like,
Well, I think what happened here was that um I was looking at the hydrophobic motif,
Mhm.
and I used that information from the passage to pick the phenol group.
so
Uh, yeah.
so let's see. So they're talking about PDK1 catalyzes the addition of phosphate to what functional group, right? And
Yeah, so when it said that in the passage that the hydrophobic motif gets phosphorylated,
okay.
I thought they were adding the phosphate group to the hydrophobic portion, which would be a phenol. No.
So uh okay. So this is kind of a pseudo discrete. So it's just saying that PDK1, which is a kinase, catalyzes the addition of phosphate to what functional group? And this is exactly what we were talking about at the beginning of the session with the phosphorus or the phosphate group getting nucleophilically attacked by the hydroxyl, right?
Hmm.
And that's basically it. So, and that also happens with, like DNA where you have like OH, like that's why you need a 3 prime end because your phosphate, backbone, you're gonna have the hydrox group connect to the phosphate backbone of the next nucleotide. So
Hmm.
yeah, so this passage like turned out not to even really need table one, which is, you know,
Yeah, I mean, like looking back on all of these questions, it seems like I was connecting too much to the passage and like looking at the wrong information. And I don't really know if that comes from me not understanding the passage the first time around or if it comes from like content gap.
so we ended up so like it was hard for me to really understand the passage as well, but it's funny because most of the things we talked about as we were reading it like ended up being what was asked. So like there was like so this question we talked about when we talked about how the hydroxyl group like so when we talk about kinases uh it's the hydroxyl group that acts as a nucleophile um and it nucleophilically attacks the phosphate. So this is exactly what we talked about. And let's see. So that's let me look at the ones before. Just let's
Do
start
you-
go back to 17.
So do you think like it's an issue with like me not understanding a high yield topic then?
Oh,
Like
okay.
if I'm trying to figure out like why this happened.
Yeah, totally. So let's see. So first, this one was
This was the cysteine
thing.
thing.
So now, so I guess that's a content thing. So now you know that uh at this pH, aka physiological pH or 7.4, um, that you're really when it comes to this, like you're gonna really look for either if it's acidic, which is gonna be aspartic acid and glutamic acid, D and E, or if it's and that'll be negatively charged, or if it's basic with lysine and arginine being positive. Histidine's weird. Histidine's actually going to be at like a neutral, like a charge of 0 at physiological pH. But those are the only ones that you need to care about.
So
So,
when histidine is given, like let's say instead of a phenylalanine, a histidine was given, it would still be 0. So, and when is it positively charged?
um, so I think the pKa of the side group is um 6, and so it would be like so that's of the side group, and then for the isoelectric point, that would be like average of 6 and like 10. So like around 8, I would say. It can be at pHs larger than 8. Um, it can be um
Well,
charged. Yep.
it would be positively charged like under oh, wait.
So,
If the pKa of the side chain is 6, then it would be positively charged under 6.
mhm.
And then for the pi, like um if the pi is 8, then if it's under 8, it would have more um
Protonation.
yes. But so then even though it has more protonation than deprotonated parts, like um it would still be it would still be 0, like at charge at pH of 7.
Oh, at 7? Yeah. Yeah. It'll be like it's a weird one because it's like there's a certain percent that is like protonated and a certain percent that's neutral. And I believe it's like either 50-50 or something like that at physiological pH. So yeah, I would say histidine to treat as neutral at physiological pH or pH, near 7. Uh
Okay.
yeah. So
Okay.
so this was just that, which I guess is like just a tiny content thing. And then this was also like a content thing. Uh
This was like it wasn't like I knew that content, but because I saw the beta decay in the passage, I chose beta decay, or I chose the beta phosphorus. So I think that wasn't a content gap. It was more of me kind of being silly.
so, I mean, I guess the reason I was saying content gap is because we went over kinases at the beginning of the session, and so, uh, like for instance, you were kind of telling me that they only like added phosphate, so that's why I told you about that, and then I told you about how they needed STY, right? And then how they needed uh well, they needed STY to act as nucleophiles, so that they can attack phosphate, and they attack the gamma phosphate. So,
Mhm.
um, so were you, because you're saying that you knew it was like the like, so like,
Once I read the question again and I realized that it was kind of like completely separate from where it was talking about beta decay, I realized like, oh, it's talking about like the order or the names of each of the phosphate groups. So then, because I knew like the gamma one was at the end. Um, so yeah, I just didn't like put two and two together the first time around, I guess.
got it. So, yep, this has to do with uh content information about kinases, and yep, let's see the next one.
This one, I
So,
think like I was reading the question wrong, or like I didn't understand what it was asking.
yeah, so this is going to be like the combination of like the amino acid thing that we saw in the first question and then the phosphorylation thing that we saw in the second question. So if they're asking, in place of this, and this just means that it's protonated, sorry, it's phosphorylated, so it has a negative charge. So we just need to find what else would have a negative charge. And this is also something that, uh, I mean, it's not specifically like content related, but a content related concept is phosphomimetic. So now you know that you'll
Hmm.
it's like phenomenon. So okay, and then the next one
I
was
think I also like I tend to be too quick to jump back to the passage and like find like what you were saying with like the shortcuts that people do. I think I'm too quick to jump back to like, oh, it's talking about like these sequences, so I need to go back to that sequence
Mhm.
in the passage. Um,
Yep. Yeah. So yeah, you want to be careful about that. And also, and then next question talks about the nucleophile part that we talked about. So like three out of like what the four or yeah questions here had like we figured like we knew like or we talked about before we even saw the questions. Like
Hmm.
this one and the other stuff like we talked about kinases and how so most of these questions like turned out to be like pseudo discrete questions and content related. So like, and even the one, so like, I know you're saying the one where it was like the gamma phosphate that it wasn't a content thing there. Um, I guess then that could be an example of, I guess, going to the passage too quickly.
Okay.
So yeah, I think it would be good to just have you kind of like read the question and kind of think about it a little bit before you go back
Okay.
to the passage. In general, like, it's better you want to be someone who doesn't have to rely too much on going back to the passage. Which means that when you read it the first time, you should have a pretty good understanding of it. But yeah, this passage was pretty tough. I mean, I still don't understand a lot of it. But they didn't ask us anything about it. So,
Mhm.
you know,
Okay. Yeah, I think maybe like when I'm on a time crunch, I don't like think about what I'm reading I mean, in the passage, yes, but like in the question too. Like,
yeah, that's like super important. So I would say you can even try to get into the habit of rephrasing the question or trying to, it's kind of like what we did with cars, like rephrase the question or uh if you can come up with your own answer,
yeah.
that can help. I mean, that doesn't work for all types of questions, but for like this type of question, yeah, it would work. And that can help you have an idea of what you're looking for in the answers, as well as making you less susceptible to the wrong answer choices.
Okay. All right. Um, guess maybe let me see what other questions I got wrong. Um, okay, so that was this passage. Oh, I guess there were some things at the end that I had some issues with. 1 second. Okay. Oh, I did not understand what they were asking at all with this question.
Um, Ratio of minimum sound intensities. Okay.
Like when it's say it because it said what is the ratio of the minimum sound intensities heard by like this group and this group. So like I didn't know like they wanted me to compare the two or like add them.
Well, what's ratio mean?
Um like div basically like division like a fraction.
Yep, yep, yep. So that's what you do with them.
But then the 64-year-old male was 20 and then 74-year-old female was 40 So I guess like I was confused because I saw the lower number as worse when actually that is better. So like it would be like a greater amount. Like you can hear more if your lowest intensity heard is 20 rather than 40.
Mhm.
Yeah, because originally I like chose 50, I think.
And how did you get that?
Um, because I did like 20 over 40 as like one half.
Mhm. Oh,
But
okay.
but like because it should be like the opposite because the male can hear more than the female, you know?
Got it. Now, what we want to do here is understand that the values given to us in the table are in decibels,
Mm.
and like decibels can measure loudness, right?
Yes.
Which is related to intensities of sound. So we want to, so we can't just keep it as the decibels, right? We want to, so what formula, um, what,
I think
decibel?
it's like D or something
Mhm.
like the intensity I over or like intensity apostrophe like over the original intensity or something like that, like I over I. Oh, okay.
Yeah. All right, what else?
Yeah. Um, re oh, log.
Mhm.
Okay.
Yep. So we want to now use this to find, so that I naught is a threshold intensity,
Hmm.
which is 10 to the negative 12. And this I up top is the one that we're, trying to solve for. Um, and so, yeah, we can do that. So let's see. So the male 20 decibels is equal to 10 log I over I naught. Um, yeah, well, let's do the math here. So what do we do?
I would divide by 10 first, to get 2 equals log of I over I naught. And then that would be 10 to, you take like the opposite of log. So it would be like 10 to the 2 on the left side equals I over I naught, which would be 100.
Um, the this ratio, yeah, but what about the intensity of this person? Let's say let's here, I'll do this. If this is for the male, so what would the intensity of the male be?
Um, you said it's 10 to the negative 12 is the um threshold.
Mhm.
Would be you would add them, so it would be 10 to the negative 10th, I think.
Yeah, good. So that's the intensity, lowest whatever, relative intensity heard by the male. Now let's do it for the female. So we would do I'll write it here, 40 decibels is equal to 10 log I female over I not. What do we do here? We get rid
Divide, so it'd be 10 to the fourth, equals I over I naught, and then you would add 10 to the fourth plus 10 to the negative 12th, which would be 10 to the negative 8.
as intensity female. Um, all right, and now we do the ratio, right? So
Mm-hmm.
we do the 1 E minimum ratio herd. So okay, so like yeah, now in terms of like do we do IM over IF or IF over IM? Since all the values are larger than 1, we would want our larger number to be on the top, I believe, right?
Yeah.
So, oops. So this would be 1e negative 8 over 1e negative 10, which is 1e negative 8 plus 10, which is E1E2, which is 100.
So even though they wrote like the sound intensities heard by like the male first, was that just kind of like to throw you off? Because like usually the ratio, if it says like the male first, you would put the male like on the left of the ratio. Um, so I guess like I was just confused why it was saying and, you know.
Yeah, so we did it like that. 1e negative 10 over 1e negative 8. We get 1e negative 10 plus 8. Wait, negative 10 minus negative 8 is negative 10 plus 8 is negative 2. And that's, 1, 2, that's 0.01. And we don't have any value like that. So,
So if they did put that, like, would that would they have to specify more like what order they wanted if they had 100 and 0.01 as an answer choice? Like there wouldn't be a way to like differentiate the two unless they made the question more clear.
um, the ratio between this and this, 0.01 versus 100, ratio. Um,
Like they probably wouldn't do that, right? Because like there wouldn't like you wouldn't be able to pick one or the other.
yeah, I don't think they would do that. And I think that the they're kind of asking us, I mean, even though they're saying ratio, which means quotient, um, it could be, I was gonna say like maybe like the absolute value or just the fact that they still have the same amount. Like, for instance, like what's the difference between 4 and 2?
Like times two.
I mean, so when I say difference, that's
Like
subtracting, right?
yeah,
So what's the difference?
two.
2, right? Now, do we do 4 minus 2 or do we do 2 minus 4? Well, if you do 2 minus 4, you'll get negative 2, but the difference is still 2.
Hmm.
So the difference here, so I think here, like the ratio here, um, like implies that it's 100, like kind of like a difference. So whether that's the male to the female being like 100th, but that can also be rephrased as the female intensity is 100 times that of the male intensity.
Okay.
So yeah, I think you're not gonna. So yeah, two things. I think that it just like the word difference, that we're really just looking for a ratio. Um, so either yeah, so either
And
the
I think like with this question, it was also a content gap, like not bringing the equation up like in my mind and just trying to compare like the actual intensities given.
yeah, but I think what you did was well, wait, you got the right.
Well, I got the right answer during the blind review because I looked at the options and like
Mhm.
that was the only one that kind of made sense because like the lowest relative intensity heard for the female is, or for the male rather, is like double the female, or like I guess I was looking at it like in percentages. So like it's 100% more than the female, you know.
So
But that might just be like looking at the question wrong.
I mean, so that's the difference in decibels. Um, but the thing with decibels is that if you double, um, or let me put it this way. So for each, so if you have 10 times the in sound intensity, that will contribute to an increase of 1 decibel.
Hmm.
So the difference, so like the difference between 40 and we could also, let's see if I can actually do it that way. So let's see, let me try doing this. Let me do something like, let's say 40 uh or 40 minus 20, 20 decibels equals to, or
Oh,
I was gonna
yeah.
do
I guess like similar
all
to
of them.
pH then in that case.
Mhm.
Okay.
Yeah, I was gonna do like the like so okay, so if I did so let's figure out how many decibels come from if you just doubled an intensity. And I was also going to show that we can, since this involves I mean, if I divide one over the other, like if I did this for male and this for like female, and I divided these, the I naught terms cancel out, right?
Mhm.
So I can really just do a direct comparison. So if I have like let's say like 2 times I let's just say like I don't know, I naught, I have I naught here. These cancel, and I get 10 times the log of 2. Log of 2 is 0.3, 10 times that is 3. So if my intensity doubles, I'm only gaining 3 decibels.
Okay.
Right? Now, what about this? If I'm doing 100 times the original intensity, if I increase the intensity by 100 times, oh, I forgot the log. By 100 times, this will be log of 100, which is what?
Uh, 2.
Mhm. And then 2 times the 10, so you'll have an increase in 20 decibels. So if you increase intensity, sound intensity by 10,
Mm.
you're going to get an increase in 10 decibels. If you increase it by a factor of 100, you'll have an increase in 20 decibels. And increase by a factor of 1,000, you'd have an increase in 30 decibels.
Okay.
So, like pH is just it's the same thing essentially, except there's no 10 term. So like for pH, it's like if you have 100 times or if you have 10 times the H plus, your pH value would go up by 1. And if you have
Mhm.
100 times, it'll go up by 10. And for decibels, since you have the 10 in the equation, if it goes up, 10 times, it's going to be an addition of 10 rather than 1.
Okay.
Yep. So, yeah. So make sure you have this formula down and you remember like the value for the threshold intensity and also that you can if you're doing a direct comparison of the two
Mhm.
you can the threshold intensities can cancel out.
Okay.
Then yeah, whether it's the male over the female or the female over the male, I think the best way would be like looking at the choices and seeing these are all values that are greater than one. So where I'm going to put the larger number on top.
Okay.
Yeah. Because I don't think that they'll give you like, something that was like, 10 to the power of -2 or something. Like,
Yeah.
I don't think they would give you that and D. So, yeah.
Okay. Then the next one. Oh, this one, I guess I just like I kept forgetting if it's outside of the focal length, like if it would be reduced versus enlarged. Like I got that it would be a virtual image, but I didn't. But I think I clicked virtual and enlarged. Um,
Okay, let me 68-year-old male, focal length of glasses, negative, right? So, okay, so
like when I saw the negative, I was thinking diverging lenses, which would be virtual and invert or virtual and upright, but I couldn't remember like if it would be smaller or bigger.
so have you seen that optics PDF I have in the PDFs folder?
I don't think so.
Okay. Yeah, I would say to take a look at it. So in it, I write that for diverging optical devices, whether they're lenses or mirrors, you'll always get SUV, small upright virtual.
Okay.
For converging, it depends on the object's distance. So, like, as the, let's say this is the object distance, and going down is getting smaller. Your image distance as you go down uh, would be getting bigger. Whoops, bigger. And since magnification is the ratio of image distance over object distance, as the object distance decreases and the image distance increases, the magnification increases as well, right?
Mm-hmm.
Now, you're gonna have so if the object is let's say like at 2F or greater than 2F, um the image distance is going to be at I believe between F and 2F. So you're going to get like small inverted real. And then as the object gets closer and closer, the image gets further and further. This can turn into same size inverted real, then large inverted real, and then like no image. Don't have to worry about that right now though. And then lastly, large upright virtual. And this large upright virtual happens only when the object is within the focal point, a focal length.
Okay.
So, all in all, what this means is if you have a converging optical device, you will always have a real image unless the object is within the focal length. at which point you will have a virtual image.
Okay.
And that otherwise you get a real image, and real images are going to be inverted. And there's one exception, which I'll mention right after. Um, but generally, yes, your virtual images will be upright and your real images will be inverted. The only uh, exception here is due to the fact that real images can be treated as objects themselves. So, like, if this is an image here, and sorry, the object here on the left, and it produces this real inverted image here,
Hmm.
real image can also act as the object of this second lens, right?
Oh, hold on. I think you paused for a second.
Oh,
Sorry. What did you say right before?
oh, do you see what I drew over here?
Yes, I see
Yeah.
it.
So the arrow, this one on the left is the object, and it makes through this lens a real inverted image here. And
Mhm.
since a real image can be a real image can be projected, it can be treated as an object also.
Hmm.
So the second lens here is going to treat this inverted real image from the first lens as an object to make another inverted real image, except this time you're inverting the inversion. So you're going to get an upright real image here. That's the only exception to the rule that real images must be inverted and virtual images must be upright.
Okay. And then for diverging lenses, it's always going to be um virtual no matter what.
Yeah, in addition to that,
Okay.
small upright virtual. So that means that if you look at the choices, so here, real and enlarged, that's converging, LIR here. Um, and that's when your image distance is like, let's say, between F and 2F, and your object distance is like, uh, I think at F, maybe something like that. Um, but okay, that can happen. Real and reduced can also happen. That's like these. Oh, sorry, not. It's like this, right? Virtual and enlarged. This can only happen if object distance is within the focal length. And then virtual and reduced will happen only if you have a diverging optical device. So since they're giving us a negative value, that means it's a diverging lens. And we know that all diverging anything uh, produces small upright virtual. That's why our answer will be virtual and reduced.
Okay.
Mhm. If you ever forget, think about like your glasses prescription. Like,
Well, I think that like honestly kind of confused me more when I was thinking about glasses because I was like, well, why would my glasses make something smaller, you know?
what do you mean when you say make something smaller?
Like make the image that I'm seeing smaller. Like wouldn't it magnify the image if I have an issue with vision?
Tell me about how it would make it smaller.
Um, well, if it's a diverging lens,
Hmm.
then it would make the image smaller.
I see what you're saying. The, I mean, then we should have a virtual image, right? And we wouldn't have it project the real image onto our retina.
Yeah.
So the thing is that although your glasses can be diverging, um, like the our eyes, the lens of our eye is still a converging lens. So the light rays still converge when they hit your eye, and you know, um, can can you know
Hmm.
come together, focus onto the retina, and then your retina takes up that information. So yeah, so the diverging lens of your glasses will just cause it to diverge a bit because you're not letting it like this over here would go like that. They diverge, and if you trace these back, you would get your virtual image here, right?
Hmm.
And your eyeballs here, and a virtual image you could only see from one side. So you see your clearer image over here, right? Um, but now this is let's say your eye. When the light rays hit your eye, they will your eye lens is converging, so they're going to converge once they hit your eye and produce that inverted real image at the back of your retina, and it's inverted. Our brains just flip it for us.
Okay.
The image that's formed projected onto the back of onto our retina is technically an inverted real image. Yeah.
Hmm.
Um, so yeah, so that's why it might appear like uh something like or sorry, yeah, so that's why you would get so you're saying wait, yeah, so that's why you should get uh your vision corrected. I think that's what you're saying, or were you saying
Yeah, yeah. Okay. Um, this one, all of these questions also I was kind of rushing through because I was running out of time. So I didn't really get to go through them super in depth. Um, this one I just messed up like how much would be left. Like I originally put 75%.
super common thing
Yeah.
that that because the MCAT's like a hyper literate exam. Like entire everything can be like the answers to things can ultimately hinge on like just the like a word.
Hmm.
So, so like, you know, right before even reading this, like I have a feeling that so something that they could ask is like, okay, how much of this product has decayed? But they could also ask how much of this product is left.
Yeah.
So that could have been it. Um, so they're saying how much is left. Um, so okay, let's see. C10 after it took the how much left is after the time it took a 75-year-old male here to walk one lap. Okay, to walk 5 laps is 200 seconds. To
So then I like divided 200 by 5
uh-huh,
to get 40.
yep.
And then, so that's two half-lives. So it would first decay by 50%, and then it would decay by 25%. So then there would be 25% left.
Yeah, so let's say, first half and half, 50-50, and then, or maybe it's easier to just do like, okay, um, like 100% to 50% in 20 seconds, and then another 20 seconds is 25%.
Mhm.
And now this is supposed to represent what's left or what's decayed.
What's left?
Hmm.
So 75% of it had decayed.
Correct.
Okay.
Yeah. So that's why, yeah, like they love to do things like that with the wording.
Definitely not helpful when they're at the end of the section.
Yep, yep. Um, yeah, if you have a patient who's like, this is like my blah 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 and stuff, and you don't want to miss out on any important info.
Mhm.
So yeah, they love to ask questions like that.
I did that one. Okay. Oh, this one we don't have to go over. I just ran out of time, so I had to click something.
Wait,
Um,
let me see that real quick.
it was just a resistance question.
So did we go over like the reason behind like did I ever draw like this?
Yeah, of the wire. Yes.
And so we talked about how then like if something's in so okay, resistance, is it proportional to length or inversely proportional to length?
It's proportional to length and inversely proportional to area.
Good. So in parallel, the equivalent resistance should be smaller than the resistors, right?
Yes, because um like there's more area for the like current to go through.
Yep. And so a reciprocal sum will always be smaller than any of the components that make up that reciprocal sum. So right away, we should be able to get rid of these,
Mm-hmm.
right? Um, and then between this and this, I guess you would have to do the math, but um, maybe I can show you like a shortcut, and like you could do like product over sum,
Okay.
like R1, R2 over R1 plus R2.
Okay.
And this could be like 6 times or 120 or E4, something like that, over adding them, which is like 80 or 8E1. So 4 goes into this 3 times, goes into this twice. So this is 1.5 E blah blah blah. And yeah, that's it right there.
Okay.
Mhm.
Okay. Then everything, this one, um,
Yeah.
I got right, but hold on, this one, and then yeah, maybe we could go through this one,
Yep.
quickly, but just so it's just saying that the hydrogen bonds are between the backbone. Like, would you be able to draw that out of like how to visualize that? Like the side chains are connecting to the backbone of the other molecule or of the other side of the protein.
Yeah. So first, I want you to tell me what's primary structure.
Primary structure is like the connection of the amino acids together by covalent peptide bonds.
Okay. And what functional group are the peptide bonds?
The the they're amides.
Okay. And what anything special about amides? Like um in terms of like their stability?
I'm gonna say they're pretty stable. Because the oxygen is partial negative and the carbon would be partial positive and then the nitrogen partial negative again. So like they have a strong bond.
So what you actually have happen here is the nitrogen has a lone pair, which
Mm.
can go here to make a double bond here, and this goes up here to make a.
Okay.
So this can give you So the amide has double bond characteristics, which makes it really strong. Good. So that's your primary structure from the amide. Now, yeah, if we had like say like or kind of bad drawing, but basically within the backbone, like the amide, or wait, that should be like the H part of the amide. So,
Oh, okay. So it's not like lone pair interactions. It's the hydrogen of the nitrogen portion, like bonding with the oxygen.
yep, hydrogen bonding. So, FON, FON, so oxygen, nitrogen in that in this case. So yeah, you can have hydrogen bonds between the backbone and not the side group. So like your beta pleated or like your alpha helix, like if you look at it like down the like if this is like the alpha helix and you look at it down this way, you're gonna see like the R groups, on the outside like that. And same thing with like the beta pleated sheets. Like the R groups are situated on the outside. So yeah, the R groups don't play a role in secondary structure, but once you get to tertiary structure, they do play a role. Like hydrogen bonds between side groups, as well as like dipole interactions and van der Waals, um, covalent disulfide bonds, and but predominantly hydrophobic interactions is what makes tertiary structure.
Okay.
Mhm.
All right. Yeah, I think that's everything then for now. Like I still have to go back and
Uh,
actually like review
okay.
more.
So let's see. So um okay. Yeah, I'm trying to think of like what we got today. Besides like just really understanding like stuff about the kinase and stuff like that. But okay. Um
Think it's like I didn't even really realize that the kinases were such a high yield topic and like that I was missing some key points. So maybe like I guess if there's other like high yield content that you would suggest like going back to like besides the main like obvious ones like amino acids or something. Like are there any specific ones that like you think are really important?
yeah, so in the shared Google Drive in the PDFs folder, I have a lot of really useful stuff. I would take a look at the optics table and
Okay.
you understand that, like particularly like because I went over like it today, like in a somewhat vague way, but if you look at the table, you will see like exactly what values of object distance correspond to what values of image distance and size. And I also have like in the PDFs folder, PDF that's just called kinase STY, and you get to see like the mechanism of that. But yeah, remember to start to make these associations, like so that when you see your hydroxyl group, think nucleophile. When you see a carbonyl group or a phosphate group, think electrophile, and like just kind of train your brain to like when you look out for that, so that when you read it and you read something with a like a whatever a hydroxyl group, you know that it's going to participate in some type of nucleophilic addition. So just train your brain to make those associations. And
Maybe I'll start like maybe I'll go through all the PDFs and like one day this week and make like Anki cards just of like those connections. That might be helpful.
yep. And yeah, and then there's also like in the shared drive like quick sheets. Like I have a Jack Weston like um cheat sheet that has a lot of really like concise, information that you can always that would probably be a good thing to like just look over like every like maybe a little bit every day to like kind of consolidate the information and knowledge that you have. Uh
Okay.
and and then yeah, also like, make sure you understand you all of like the equations and stuff. Like
Yeah,
and
I'm still like getting it. I think something that might be helpful that I was talking to like one of my friends about is like in passages highlighting the units in the passages specifically so that even if I don't understand the equations, I can try and like piece together like what units they're using.
yep,
Uh,
exactly. Like, yeah, I always say if you don't know the equations, the units will help you. If you don't know the units, the equation will help you. So, being very nimble with the dimensional analysis and stuff is going to really work to your advantage. So, yeah, so I think that's what we can do for next time. So let's see if we can get um okay, yeah. So your next test is
I think this Friday.
this
And then
Friday, the 7th.
yes, taking it Friday and then our next session is Tuesday or the following Tuesday.
Let's see. Oh, the 11th?
Yeah.
Okay. If you want to meet like this week, I have an opening too, if you want that.
Um, what do you have an opening on the 6th or the 5th, maybe?
I have one on just the Friday.
Oh, okay. Um,
Yeah,
what time? Because I'll be taking the exam, which would probably finish around like 3:34.
yeah, that could work. Yeah, because I have a spot open at like 3:30.
Okay. I
Cool.
might be like a few minutes late,
Oh
just
yeah.
like depending on the test.